The mechanism of the
Mechanism of the
pathogenesis of
pathogenesis of
influenza
influenza virus.
virus. The genome of
The
the
genome of the influenza virus consists of 8 single negative stranded RNA, and each RNA segment encodes 1 to 2 proteins. The, the negative sense RNA cannot be directly translated. So the positive strand must be synthesized from the negative strand, and from the positive sense RNA, it is translated to produce the viral RNA, viral proteins. The most important envelope proteins required for the pathogenesis of the influenza virus are hemagglutinin, which is required for the entry of the virus, and neuraminidase is required for the release of the virus particle from the host cell. The first step of the pathogenesis is the binding of hemagglutinin through its hydrophobic SA2 domain to sialic acid on the host cell surface, and that leads to receptor-mediated endocytosis of the virion. And then M2 ion channel pump proton to the endosomes, which decreases its pH, and at around pH 6, conformational change of the hemagglutinin occurs, and this conformational change leads to binding of the hydrophobic fusion peptide to the plasma membrane, and then proteolytic cleavage of the hemagglutinin to SA2 and SA1 by the soluble host protein or host cell proteases results fusion of the viral envelope and plasma membrane. And then the virion releases its genomic negative sense RNA with the RNA-dependent RNA polymerase to the nucleus of the infected or host cell. The positive sense RNA is then synthesized by the RNA-dependent RNA polymerase, which is then used to synthesize the negative sense viral genomic RNA. And the virion positive sense RNA hijacks the cellular machinery, that is the ribosome, amino acid tRNA, to synthesize viral proteins, that is hemagglutinin, neuraminidase, M2, capsid protein, etc., which are assembled to new virion particles under the host cell membrane and released through sialic acid residues, sialic acid receptor mediated budding process, that is receptor mediated exocytosis. And the release required cleavage of the bond between the sialic acid and the hemagglutinin, and this is done by neuraminidase.